Hi, Dr. Miyadera. Good afternoon. My name is Esha, and I'm a high school student interested in being a veterinarian. I'm so excited to welcome you to the interview series on the careers of veterinarians. Thank you so much for joining us. Let me start with an introduction of our speaker. Our speaker today is Dr. Miyadera, a board-certified veterinary ophthalmologist and an assistant professor at the University of Pennsylvania School of Veterinary Medicine. Born and raised in the southern islands of Japan, she considered herself a country girl until she headed to Tokyo to attend high school and university. While at the University of Tokyo, she intended to pursue genetic research, but when she learned that the school had a veterinary program, she thought it might be more fun to work with animals and went on to obtain her Doctor of Veterinary Medicine from the University of Tokyo, Japan in 2005. After receiving her DVM, Dr. Miyadera did an internship in Tokyo and got her first dog, who was a hemophiliac and would bleed sporadically. Dr. Miyadera found herself constantly tracking down transfusions for her beloved dog and learned the difficulties of owning a dog with a genetic condition. This strengthened her interest in genetic research and led her to study a genetic disease during her internship. She focused on a gene mutation that was making dachshund dogs blind and started working with an ophthalmologist and taking blood samples from the dogs she saw. Learning that this blindness was originally seen in England before the breed was imported to Japan, she followed the disease and its research to the University of Cambridge in UK to complete her PhD on progressive retinal atrophy, and she was able to use the data and samples obtained during her internship. After her PhD, Dr. Miyadera completed a post-doctoral research and residency in ophthalmology from the University of Pennsylvania in 2015. At Penn, she sees patients and pursues her research, and her background allows her to see the disease from both the clinical angle and the genetic angle. Her clinical specialty is inherited eye disease in animal, and her research is focused on the molecular basis of inherited retinal degeneration in dogs. She currently lives in Pottstown, Pennsylvania with her family of 4 humans, a tuxedo cat, and a Portuguese water dog or beagle mix puppy who is joining the pack soon. Thanks again for chatting with me today. With this introduction, let me extend a warm welcome and thank you to Dr. Miyadera for chatting with me today.
Wonderful. Thank you, Esha. So that was an excellent summary, and I guess you already have most of my story today. That was an excellent summary, and yes, thank you for the invitation. Yeah, I'm very excited to chat with you and share my stories.
So first, could you tell us about the type of work that you do? Feel free to share your screen and show any pictures or slides if you want to.
Sure, let's share this screen. Yeah, so I have a PowerPoint, which I would make it full screen, maybe. Can you see the screen? Oops.
Yes.
Okay, perfect. All right, so yeah, I've put a summary. So yes, I would be an example of a veterinarian and who's called, we like to call ourselves clinician scientists. So we kind of are on both sides of different worlds in the clinics and also as a scientist in the research area. And also specifically, I'm in academia, so I work at university. In fact, I've never left university. This is fortunate or not. We will see. But so I as in your introduction, after finishing University of Tokyo Veterinary School, I went on to finish my PhD. That was in England, and after that, it was a postdoc here at UPenn. And ever since I have not left Penn, it has been 12 years. So yeah, this is a very academic career path so far. So let's see. But first, your question was, what do I do? So what do I do? So many things. So as I said, yes, I live on different worlds. Maybe I'll close this one. Okay. So yes, one hat I wear is as an animal doctor, eye doctor. So I work in Philadelphia, where we have the University of Pennsylvania Small Animal Hospital. And this is a big referral hospital with all the specialty services, including ophthalmology, neurology, dermatology, dentistry, you name it. and I work in the ophthalmology department. It is a full-time service, so we are open 5 days a week, and we do cover weekends. So say if your dog runs into a bush and the eyes slam shut, something's wrong, people would walk in with a dog, and one of us will be seeing the patient. But yeah, as an animal eye doctor, so this would be our eye exam room, speaking directly with the patients, patients, and then the clients. I do, as during the service hours, I teach students and also residents. So residents would be veterinarians who are working towards specializing in a certain subject. So ophthalmology residents would be the future veterinary ophthalmologists. So I train them as well. And we won't perhaps go into too much of eye exam and everything, which is another fun area to talk about. But we use instruments that are mostly all of them are what's used in human ophthalmology. This specifically is used in human pediatric ophthalmology, that we can hold and move around as our patients, as they tend to move around a lot. But yes, we have good help. So in fact, perhaps a lot you can say with veterinary clinical work is that it's not something you just walk in and you can do on your own, like perhaps you would get with your primary care physician working in the office. We always need someone's help holding the dog, assisting us. So perhaps that's one unique feature. But so that's one thing I do as an eye doctor. And just a note about Penn Vet. So this is a small animal vet hospital. We do have a large animal vet hospital out at in Kennett Square, New Bolton Center. This is about an hour drive from Philadelphia westward. And there we do our large animals, horses, food animals. And during my residency training, I had the opportunity to work with those very fascinating larger animals as well. But for the most part, right now, I'm focusing on small animal clinical ophthalmology. And these are the type of eyes, just an example of some eye diseases I might see. So if this is your eyeball from front, cornea, that's a cornea disease. And then you might have your lens disease, that's a cataract. All the way to the back of the eye, you have the light sensing photoreceptors containing retina. That's a retinal disease. All of these I deal with. And perhaps that's another point of veterinarian versus human medical ophthalmologist is it's specialized subject, but we still, perhaps there are not many of us enough, or that perhaps due to the number of patients we see, which is not comparable to the human area, we do see everything, which perhaps so we don't have a retina surgeon or specialist or cornea specialist. We are mostly general ophthalmologists with some people may have special interest, but still see everything. So in a way, we might have a little bit difficulty spending enough time in one area of the eye as deep as a human ophthalmologist would do. But on the other hand, we get the whole picture and we can treat the whole eye, which is, it's very rewarding in a way. So that's just an example. Another thing I do, so I work also clinically outside of our small animal hospital, and this becomes part of my research area. And as a clinician scientist, in fact, most of the work is on the science side, and I have more commitment on the science research. And this starts to become more research. And it still has the clinical aspect, but I am doing what you see here in these pictures. So these are, here I'm going to dog shows, and this is like a hotel room. We were stuck in a camping car for 3 days in the dark, just examining dog after dog. And if you go to a dog show, you see And in a specific breed dog show, you see the same breed, several hundred of them at a time. And as you see a lot of them like back to back, you start to see the trends about what's the issue eye-wise in that breed. And the ones I've been going to is that we have a problem that the breed club would like me to address, and I'm working with their we are working collaboratively with them. So that's one thing that where I start my clinical research. This is where I go and collect my data. And here to the right, it's a similar situation. Again, there is an issue in the breed. There is people want me to look into it, and I go, and this is specifically going into people's houses in their, I think this is a bathroom, just a living room. But they are, until I started working in these settings, I did not know there were interest and willingness from ordinary people, like, I like to call it citizen science, but people are very willing to come together and help me find out, solve issues in a breed. So that's one. And then, so this is still, these are also dogs coming from a breeder who thinks there is an issue, or in the breed, people know there is something that needs to look at. And I'll have these dogs come into our facilities, and what's not available on a dog show, but we do have cutting edge instruments that we can study their eye function, that's a function test, or eye structure in detail. And this gets very exciting that we finally get some numbers, or we can define a disease that people with prior to that only anecdotally knew or thought that there could be something, some process might be happening, but we can start moving into properly characterizing what we like to call phenotype in the genetics area. And I snuck in here, so to learn, I study eye disease in dogs to learn about vision or eyeball, using the dogs or in the dogs, but also in people. That's also what I'm interested in. And why am I putting in people here? Because so a lot of us who does clinical research in the veterinary field, we also often work with our counterparts in human medicine. And that's because we see a lot of things in common between our veterinary patients and the human patients. And I guess this is one way to look at it, the organs. And there are also other animal species, something like mice or even zebrafish, that basic scientists have been looking closely at to see if we can get a clue about understanding people's diseases. But dogs, unlike zebrafish, say, there are a lot of things that's common, the organ structures, how they operate. So they become very much a good model. And another good word people like to use, translational. What we find out in the dog can be immediately translated to understanding or even treating human diseases. And I put this here. These are the key. And Esha, let me know if this is timing is okay. But you can stop me anytime, but I will keep going if not. So these papers, particularly this Nature paper 2005, came out when I was a veterinary student. And this was very exciting. Until this point, we were starting to learn about dog human genes that works in the different parts of the body, leading to the specific function. In dogs, it was not quite well known, but once the entire dog genome, this is about 3 billion DNA nucleotides, A, T, and C, and G, they all got read for the most part. and was put out on the database that everyone can access. And once that came out, learning about what molecules, which could be your gene or protein, which of them links to certain disease, or which of them, when they go bad, leads to disease, that detective discovery process become facilitated, let's say. So this, perhaps thinking of my career path, these developments were definitely the key in further moving me towards working in genetics, veterinary genetics, and then ophthalmology specifically. And talking about, again, why use dog as models to, and how can we say we can understand this in people? To the right, yes, we share the environment. That's another factor that can trigger or modulate your disease. So that's something that's shared between dog and man. Genetics, I'd like to look at genetics, but dog, in fact, is a very good population, species to look at if you want to find what gene or genetic variant is causing a disease or phenotype, because they're quite different if you look at across the dog population. So their genome or the genetic makeup is quite different, and a lot of different variants would come up in the different breeds. But then if you look at one single breed, they have been bred in the past, say, century or two, and very narrowed down with a bottleneck and then expanded. And their genetic makeup is very uniform, say in this beagle population or any other pedigree, so-called breed. And so if a disease comes up in this beagle but not in the other, if you compare the genetics of this affected dog and an unaffected, it's much more straightforward to find what's the difference is than trying to do that in the human, the very mixed, heterogeneous, we say, but a very more variable population. So that's another thing that's key, why dogs come into this kind of picture. So I guess this could be getting towards the end of what I do. But yes, I talked about a little about genetics is part of my research interest. And once I have a disease, I have a population of dogs that I'm interested. Say I have a group of dogs that goes night blind, while another group of dogs that ideally are related to these dogs, but they are perfectly normal. And they look genetic in its cause. So then I would like to find what gene is doing that, because maybe I can treat them once I know what's the disease mechanism. And what I do is do a gene, I call gene mapping, or using technologies that allows me to find where in the dog genome, which chromosome, and then eventually in which gene might the cause be in that's making certain population of dogs blind. This is another technology I use. This is whole genome sequencing. What took for the human genome over 10 years, and for the dog genomes years to decode the 3 billion base pair, now you can do in weeks and at much less cost. So that is one tool I use to try to get to the bottom of the genetics of diseases. This is just an example of a mutation I found that was a deletion of one base pair, affecting the splicing and expression of the protein. So this, and that was making that group of dogs blind. And finally, so yes, we have a gene that now we know that makes a certain population of dogs blind. And in fact, in the previous story that we found a blinding gene mutation, the same gene in people when it is mutated, it has been known to cause night blindness in people. So again, that translational aspect is very key here. But so those dogs were born night blind, and they would be night blind for the rest of their lives. And there had been no treatment up to this point, but we did find therapy for them. And the last part is, once you know what gene is not has a mutation, you can fix them. And there are different ways of fixing a dysfunctional gene and its product. But so this is one example. And in fact, I have two examples. And this is the first one, that we're trying to fix a disease in the front of the eye. So that's your cornea. And this is a storage disease. That's also, we see children patients with this condition. But your cornea, the front window, becomes cloudy from the disease. And we knew which gene was causing this disease. So we put a correct version of the gene in a virus, and this is a non-harmful virus that will just only be helpful in delivering your correct version of a gene to where you want to treat. We ended up injecting that, now you can call this a drug, to the front of the eye. And this, we managed to make this treated eye more clear. So you can see all the way to the back of the eye. By the way, this is how the back of a dog eye looks like: the retina, the optic nerve head, and retinal vessels. But so, yeah, so this is something that would be the ultimate goal of all what I do, starting from the clinic part, characterizing the disease, finding the genetic basis, and then targeting what I found from the genetics to make that as a therapy target. And in fact, actually test them and achieve the goal. And that's perhaps my ultimate goal. But from here, now the human ophthalmologists are going to take over and move things on to clinical trials in patients of the same type of disease. And I have another, so this is the last but second example of a therapy that we developed is, now these dogs had issues in their disease in their retina at the back of the eye, and these tissue needed the correct version of the gene to be delivered. And we ended up going inside the eye all the way to the back, and then making a little balloon just like this one here under your retina. And here, this balloon contains the drug, which is the virus containing the correct version of the gene. And this dog in the treated eye, this dog was bumping into this object in the dark or in dim light, but now he could see completely in the treated eye. So that was, again, a very promising and rewarding project I have been working on. So I think, yeah, this is just a summary. But yeah, all of this, it's, as a clinician, this would be this part. I see the patient, encounter some key conditions that can be helpful in treating people or treating themselves more efficiently by studying what gene is involved and how we can target them. And in the meantime, we can also study what exactly those genes are doing. That becomes more basic biology, but this is something we are also interested in working on. So I think that would be my brief or long story of what I do.
Thank you so much for sharing all about your work with us. That was really exciting. I was wondering if you could tell us a little bit more about your education and training that prepared you for this career, how it helped you, what were the prerequisites, where did you study, and if you had any influential mentors.
Okay, mentors, right. So let's pick on. I have a few slides, I think. And really, a lot of my journey you touched on nicely in the introduction. That was, again, very helpful. You see a full screen?
Yes.
Yes, okay, great. So, and then maybe the title is covered, is it?
Nope, I can see
No?
it.
Okay, oh, great. So yeah, oh, good. Yeah, so again, this is where I started my journey even before vet school. But so I am from the South Island of Kyushu in Japan. And I did not personally grow up with dogs and cats. Now I have a cat and we're expecting a puppy. But I grew up, in fact, with chickens and hens, one at a time. And we would go and grab a retired hen from our local farms that were provided to the local community, and we would bring them home. And we had a little chicken coop in our house. I was the one to collect the eggs. But my veterinary exposure growing up was limited, maybe some fish. And I was, in fact, afraid of dogs. There were some mean dogs in my neighborhood, so I was always avoiding them. So yeah, it was not until vet school that I finally got some very good grips with mean dogs. But yes, so I studied there, went to Tokyo for veterinary school, high school and vet school. Vet school in Japan is 6 years right after high school. The final 3 years of vet school, you are involved in a bachelor's research program, and you would be writing a thesis. I picked up, of the few possible options that I could have taken, I picked up this topic, coagulopathy in Japanese black cattle. Some people might know of Wagyu. And these are the population of cattle that you see a lot of them in my hometown, not in the neighborhood, but in the area where there's the industry. And as part of my bachelor's thesis, it was going to be opportunities for me to do field trips, visiting farms, collecting blood samples to examine this coagulopathy in the Japanese black cattle. And that was the primary reason I first ran into an inherited disease. And coagulopathy is but the type we were looking at was an inherited hereditary form. And there were more, so what farmers were saying is, so they would have their male animal castrated, and the bleeding from the castration site never stops. Or they would do an ear punch in the young animal, and there was some abnormal bleeding from there. And that's how it was initially discovered. But it turned out there were more than 3 different hereditary types of bleeding disorders. This included a certain type of hemophilia that's also relatively well known in people. But so all of that was coming from an extreme inbreeding of these very special cattle to get this perfect marbling in your beef. And up to some very popular sire would have 10,000 calves out of himself, and by artificial insemination. And in that way, there was, in the coagulopathies that we're seeing, was a direct result of such extreme inbreeding. So this is how I initially stumbled across, but I really found it fascinating to see how people get in and regulate the breeding, and how that affects directly how disease manifests, show up in the population, and how things can spread pretty quickly within generation or two. And I wanted to continue studying inherited diseases in animals. And so after I finished my bachelor's studies at the University of Tokyo, I did enroll in a PhD program at the University of Tokyo and started looking into what I might work, because now I'm more based in the small animal hospital as a PhD student. And this is prior to me moving to Cambridge, but I spent 2 years, a little less than 2 years in Tokyo looking into this situation. In fact, so this is, again, when I was a vet student, the population of dogs around you were just looking very strange. Every other dog, almost, it felt, walking into the clinic were miniature long-haired dachshunds, because there was a huge demand and popularity of this breed. So we were seeing a ton of this breed, which had not been as popular just a decade ago. So you can imagine the high rate of inbreeding, like a dad being bred to their daughters, and so that they can quickly make a lot of puppies from a limited number of breeding dogs. But so as a result, I did stumble across an inherited disease in this breed, in the dachshunds. And it was a blinding disease called progressive retinal atrophy. And this is, again, when we look through their pupil to the back of the eye, this is how your retina looks like in your retinal exam. This is a normal retina. With this disease, the retina becomes thinner and thinner and thinner over time. And the changes you see through your retina exam is like this: the vessels, blood vessels becoming narrower, and then the back of the eye looking more shiny, looking shinier. And patients, by the time they come in, prompted by them walking, for the clients, they would notice their dogs running into everything. And by then, their pupils are wide open. But so this is the disease. And I started contacting breeders, clients, owners, and started visiting their house, just like I do right now. But yeah, went there to their place, did an eye exam. This was helped by my then an ophthalmologist at University of Tokyo colleague, Dr. Kato. But so she did all the eye exams for me. I collect the blood. I held the dog, perhaps, back then. And now I really wanted to find out what this is caused. And as I collect, so these dogs were all related, belonging to a breeder. And I would write pedigrees. Okay, this mom, this dad, these puppies. In 2 of the 5 litters went blind. The other did not go blind, and so on. And it looked like it definitely runs in families. And you can figure out the mode of inheritance. Is this recessive, dominant, or X-linked? Those things. So I started working on those phenotype aspect. But then, yes, so halfway into that work, a paper came out and telling me that, in fact, someone in England, in the UK, has figured out what makes the dachshunds blind. And they found a mutation in, oh, this is a typo, but RP-GRIP1 gene. Maybe this is from a different talk, but maybe I won't go into too much details. But yes, so number 1 here, it looked like someone already solved the issue. Nevertheless, I looked into the dachshunds in Japan, and I did find something a little bit different from what they had found in the UK. So I thought, and it looked like it was worth looking into it. So this, in fact, by this point, I had moved to Cambridge. So I visited the university, and there was a veterinary geneticist, Dr. David Sagan, who would become my PhD supervisor. And I spoke with him, expressed my interest in pursuing veterinary genetics, and he kindly accepted me as a PhD student. So I restarted my PhD at Cambridge. These people, Dr. Catherine Mellish, Dr. Keith Barnett, they were the people from the group at the Animal Health Trust in the UK who found this genetic variant. But she became my PhD advisor and has been Dr. Sagan and Dr. Mellish. Both those two have perhaps been one of the strong, influential mentors that really trained me in genetics and to look into the phenotypes closely, and also what tools that would be appropriate in looking into the genetics of disease. Yes, and eventually I did find out some factor, or we call modifier, that would explain there was some discordance that I was observing in the Japanese dachshund population. But we did a genome scan using this DNA chip and found that there were some dogs that were going blind very early versus dogs that were still sighted, that had normal vision at certain age. And I found a genetic pattern. This is what it is in the former group. So that was the essence of my PhD work. And this is the building I worked through my PhD, 5 years, and the lab I worked at. Dr. Sagan, my PhD supervisor, and all the lab mates, people in this wing of the lab has been very helpful in teaching me basic molecular biology, Western blotting, PCR, polymerase chain reactions. Those are very basic and commonly used technique, but perhaps something that are not as rigorously taught or trained. I guess as a veterinarian, you would not need those molecular biology skills, but during this phase of my training, I was able to acquire those skills and knowledge. So definitely a very important key phase of my career development. And then after PhD, what shall we do? And there are many choices when people finish their PhD. Many people go on to do a post-doctoral fellowship or postdoctoral research position. And yes, if you're very into research, but there are different places you can do research. Academia is one, perhaps industry is another other place, and other places perhaps that I can't describe right as yet. But yeah, so people who are still thinking perhaps postdoctoral research position is always a good idea to enforce your research experience, because you just got minted as a fresh PhD person, but you only have several years of experience. By doing a postdoctoral position, you perhaps get your own research project and become a little bit more independent than a PhD student would be, but still under a mentor. So that's what I did. I found an excellent mentor here in Philadelphia, USA. And so I don't have a picture, but Professor Gustavo Aguirre was a big, well-known and established figure in veterinary ophthalmology and specifically in retinal research. And he has characterized genetically and as well as phenotypically many eye and specifically retinal diseases in dogs. So I contacted him and was fortunate enough to squeeze myself in as a postdoc in his lab, and I moved to Philadelphia. And so, yeah, so it turns out Philadelphia is an excellent place to do vision research. So I'm here at the vet school. That is New Bolton Center, where we do the large animal. And then next door, you have the Penn Medicine. We have there is the Shea Eye Institute that is that to cutting edge eye research. And you also have the industry right there. Some of them had directly spun off from research from the academia. And perhaps this could be, if you're thinking about veterinary school in the US, University of Pennsylvania is one of the few, if not the only school that's vet school right in the heart of a city and within walking distance from all the other, the medical campus and the industry partners. So this is a very good environment to work and collaborate on research. But I came to Philly. I did the postdoctoral research fellowship for 1.5 years. During this time, there emerged an opportunity for me to pursue a clinical ophthalmology residency. I came to the US just focusing with an idea that I will be focusing on molecular biology, genetics. But and I happened to be working on ophthalmic disease, but there was an opportunity here, and it was perhaps the single best thing that happened during my career development, to be able to get into a veterinary ophthalmology residency. And that was a 3-year residency, and you work all aspects of eye disease, from medical treatment, surgical, pathology, immunology. So you are now well-rounded clinician ophthalmologist. And after the 3 years, now again I have a choice. I can even go back to Japan. I could move on somewhere. But again, I was fortunate to be able to stay on as a faculty at UPenn and doing exactly what I've been working on. So I've worked on honing my molecular biology skills, and I have a strong clinical training in ophthalmology. So combining those two is the kind of work you saw earlier, that what I do now with both the genetics and clinical background. I think I'll stop there.
That was a remarkable journey. Thank you so much for sharing that. May I ask, what are some of the most rewarding aspects of your career?
Yes, I have it. So, well, there's a lot of things, and there are rewarding things as a clinician and as a scientist. And yeah, both are, or all of them, I cannot put numbers to it. But yeah, in the clinics, so in one word, it would be people. The most rewarding thing is the people. So you would think one would go to vet school because you love animals. And of course, I'm interested. I like animals. I love spending time with them. But the greatest joy comes from the people I interact and work with. And so this is Mr. John Wagner that I met through, was a client of mine. He had his dog Sheba, who is his service dog. So Mr. Wagner, he's not blind. His vision is okay, but Sheba is not a vision guide dog. Sheba is a diabetic alert dog. And when Mr. Wagner's blood level will go down, she would stare at him and let him know that she needs to eat something or to deal with it. So Sheba was John's precious service dog that they would be together all the time. But one day at a routine eye exam where he went, the veterinarian who saw Sheba said, there is something odd about Sheba's eyes, and I think we should refer you to Penn. So she came to us, and we saw, it's a little bit hard to see, but a little bit of pimple, a dot, which turned out to be a type of tumor growing on her iris. That's the brown tissue. And we did a an high resolution ultrasound of her eyeball. This is the front window of her eye. And this growth here is a little dot over there. But so John has been coming to see me, I don't know, 5 years annually. And so each time we have a long discussion of, okay, what shall we do with this little pimple? And in fact, you don't have to jump and try to take it out, because as an ophthalmologist, we know, and having done pathology or having studied all the literature, we do know that this is not gonna kill her. There are certain tumors that can affect the life of the individual, but we do know this can be locally invasive, but it will not metastasize, so-called. So we had the long discussion. I gave him the options. You can potentially cut them out, it's possible, or we can do a little bit of laser and debulk, make this grow smaller so that it won't grow too quickly. And after some years of thinking and long discussions, he decided to go ahead and do laser. There was we were reluctant to put her through anesthesia, because she is a highly trained, highly attached, very precious dog. So we didn't go into surgery the first year, but eventually we felt that it's time. So we did that, and we were able to shrink this tumor. And over the following years, it gradually started growing again, but it was within expected. And Shiba has still keeps her eye and doing great. But yeah, so I guess I just told a story of a case here. I guess there was not a climactic moment that, okay, this was the most rewarding, but all these interactions and going through, looking in their eyes and feeling that they're comfortable, like they understand and following all the options I'm presenting, and they're comfortable having spoken with me, that they've come to a specialist, and they are confident with their decision, and they can go happily, or they can forget about this for the next year until they see me again. And all of that that I'm able to provide, that service has been a great rewarding experience.
That was a
And
really
thank you, Esha. And one more thing, so that's in my clinics. And
mhm.
what did I have in the, yeah, so on the research side, I told that I spoke that in the clinics, you can't do anything by yourself. In research, it's the same thing. And do any work, you need a group of people, and each of these people have very specialized skills on the bench or working with animals. And each of them, yeah, I worked very closely and being helped by that. And we together made the achievement of treating blindness or being able to come up with a therapy is, yeah, again, that's the biggest part. The teamwork aspect has been the biggest reward. Yeah, that's all for this
Thank,
part.
thank you for sharing that. Now, could you tell us a little bit about how you believe the profession has changed during the span of your career, and how you and other successful vets have adapted to this?
Right. And yeah, certainly. So I became a vet in 2005. So it's not the longest career, but certainly there have been developments, including what we've already talked about a little bit, which is technology. Yeah, so it would be more in the research aspect, but for sure, this came out at the beginning of my veterinary career, but the finding from these and then the technological development has had a impact in the changes of how we work on things in the lab, and as a result in the clinic. But yeah, and even within this slide, the top half is less utilized at this time. So the DNA chip, so these are a DNA chip that would type, let's say, up to 700,000 markers across your whole genome. And this would point you to where are the difference between your blind dogs and non-blind dogs. That's the precise chip I used. But this technology was shining and cutting edge back then, but now this is now being replaced with whole genome sequencing. Things have gotten faster and more affordable. So trying to adopt that is definitely a challenge. So you feel like you did a PhD, you've accomplished a skill. This is a skill I am going to carry forward, but that wouldn't necessarily be the case. And I found that you need to keep relearning to catch up with the development. And even to the right-hand side, this is, didn't talk much about, but DNA test is something that is utilized more in clinical veterinary clinics as well. Certain conditions, you can check their genes to see if this certain dog is predisposed to certain disease. And so this Optigen is one company that was highly used in the past 2 decades. But now they would test one mutation at a time. Again, this is now replaced by a more comprehensive testing, such as these ones, and they can test several hundred different gene mutations with one swab that you can mail in. So all of that development, you need to be on your toe, keeping a watch and learning all the time to be catching up. One thing I wanted to mention also is in the science side. So once you become a PI, so principal investigator, you run a lab, which I do now, so you need to go apply for funding so that you can do all the exciting research and test all the interesting ideas you have. But you do need to be going and talking to people, writing grants, papers in order to get the money and get the people to do that. So yeah, writing is something that I feel perhaps I didn't necessarily learn in vet school, but something I learned myself or acquired or took courses. And maybe this could be a little bit later, this part. But if I focus on something that's new, I realized some PIs or researchers, they are veterinarians, but now they finding themselves in a position that they have to start a company because they have this amazing idea and that leads to therapy, and there is a needs, there are demands and people who would pay for it for the person to do it. And to establish and run a company, some veterinarians are trying to do this, but and this is I feel it's a new development rather, and say startup, you would say. And yeah, that's something maybe some people can quickly catch up. Maybe another avenue is you work with some people who are good at running a company. But yeah, that's something I realized perhaps even in the past 10 years since I started a faculty here, that have become something that's more, I see common. Okay, I stop there. Thank you.
Okay, so thank you for sharing your insights. Is there anything you wish you knew while you were in vet school about other career paths?
Yeah, in fact, yeah, I'm very glad that you are talking to people this way. I think this is a really good occasion. And yeah, definitely I don't think, I wish I knew more about the other career path in general. And being in University of Tokyo, being an academic, strong university, research path seemed more feasible. But perhaps I feel as a veterinarian, when you choose research, it seemed like you need to now go lock yourself in a lab. Or if you do choose clinical medicine, maybe you're going to go and out on the clinic floor all the time. But the option that there is an option to become a clinician scientist so that you can actually do both is something, yeah, I figured it out, and perhaps I knew a little bit, but it would have been nice to speak with those people and get more exposure. And perhaps, yeah, anything I knew related to that, yeah, I guess it sounds like a great idea, clinician scientist. You can do both. You can speak both language. You can contribute to both worlds. It's a great idea, but perhaps it's not for everyone, or maybe it could be for many people, but one should understand that as a clinician, I'm never going to do as great a cataract surgery as my colleague here at the vet school ophthalmology, who's doing cataract surgery every week, because I'm doing all the other research. But, and same thing for the research, I may not be as up to date with all the research paper as my PhD colleague who's doing research 100%. So yeah, so at one point you realize that and you learn to live with it. But up until then, you feel you are less than these people who are committing their time 100%. So yeah, I wish I knew it. I had understood that better going into the clinician scientist path.
Yeah, that's really interesting and helpful to know. So, kind of building off of that, how should a vet student go about making choices about their career paths after vet school? And do you think that vet schools prepare vet students for this?
Right. So yeah, how should they go about? Yeah. And yeah, I personally think there's nothing will go to waste. And I, for one, I feel I was not committed. I had an interest, but I was not committed. And maybe, yeah, I was perhaps more flexible, to put it in a positive way. To put it negative, maybe I was not as determined, but I was flexible and I saw opportunities and I took it. I said yes to what I thought struck my interest. So be flexible and yeah, go, yeah. And and yeah, I had a note saying, yeah, I think it's good if you have choices even, but and then if you had, or if you were able to try different places, I think it is important to find a place where you are valued, and you can keep asking you once regularly, do I see, am I valued? And they will be. If not, yeah, you can always, don't be afraid to change paths, change places. It's also good to find a niche that your strength, that you can say, oh, I can provide this different from other candidates. I found my niche of genetics, which was still not as maybe it was becoming sexy, but not many people were doing it back then. And I had my own population research niche that I grabbed and held onto it, dragged it to the UK and now back to the US, or here to the US, and still working on a very particular disease that I call claim ownership. And you asked about would vet school prepare vet students for this? Yeah, and yeah, I haven't studied at the American vet school, but maybe okay. I feel there is a lot of still, though, might be a bit more cuddling going on than necessary. So we let's see. There's a lot of care taken not to hurt vet students' feelings, and but I think, yeah, hopefully one would come in being prepared to take criticisms and and learn from that experience, or being instructed otherwise. And I guess that's one thing I thought I would point out.
That was really great advice and that was really helpful. So I have one last question. What do you think are some of the common mistakes early career veterinarians make?
Right. Yeah, and I talked about being flexible, which is positive and that has its downsides. But I think from what I have experienced is there are some early career veterinarians that is determined. Determination is a good thing, and a lot of people get get to places thanks to that determination. So by that I mean someone who thinks, oh, I I must become an ophthalmologist, which I wholeheartedly uh, cheer on. But unfortunately, there are not enough ophthalmology residency positions. Not everyone can be what they want, and sometimes certain area looks more, I guess, sexy than others. And but I think it would be a great skill to learn to, um, like open your new area, or to find joy or find meaning in certain areas. And of course, this is not to say, well, just let people walk all over you and you just get the last piece and be happy with it. It's not that. You do your best in everything you do, and I guess hope for the best, but be flexible when a different wind starts blowing in a different direction, or something like that, I guess.
I, Dr. Miyadera, I really appreciate you taking the time to chat with me today. Thank you so much for sharing your incredible career journey, and I wish you continued success.